All right, thank you for the introduction. So mathematical models of protein regulatory networks are very powerful and widely used approaches to investigate cellular processes. Here, we construct the wiring diagram that represents important reactions and influences in the control system, and then we develop biochemical. Then we look at the biochemical mechanisms in the system and solve the dynamical equations to predict the time course of every component in the system with response to a specific stimuli. A model can use many approaches. In this figure, you can see an ODE-based model, and yesterday from one of our keynote speakers, we have looked at an ODE-based model, a Boolean model. However, there are many other types of models that exist. They can be rule-based or based on logical formulae. The power of a mathematical model really lies in its ability to predict the state of the system under different perturbations. So in a typical modeling cycle, we begin by building a model, and then we simulate this model under some of the previously unstudied environments, and then we make certain predictions. We then go ahead and validate these predictions by experimentally, and then we may reconcile any differences from the model predictions and experimental data. We can repeat this process over and over, hopefully improving the model and our understanding of the underlying biology. So as models get larger and larger, there's a potential to dramatically improve this cycle. We can simulate this model under all single mutants, for example, all double mutants, triple mutants, quadruple mutants. That's a lot of combinations right there. And we can then generate a lot of these predictions, but we'll then face two major challenges. The first one is we have these large number of combinations that we seek to make predictions for, and even if we somehow manage to make predictions for all these large number of combinations, it's almost impossible to manually plan experiments to make and characterize these hundreds and thousands of potentially interesting predictions. So the solution we propose is to develop methods that can analyze all simulations to determine which might be informative with respect to our model. And more importantly, we seek to design algorithms that can create plans on how to perform these experiments in wet lab to test these predictions. So ideally, these algorithms should maximize the number of strains we can test while keeping the budgetary constraints in mind. So the primary focus of our work is a novel approach to synthesize experimental plans. So most of my talk, I'll discuss our algorithms cross-plan. But first, let me quickly tell you how we compute potentially informative predictions from tens and thousands of model simulations. So as a test case, we used this ODE-based Budingay cell cycle model. This model contains 59 species, various proteins in their modified forms and complexes. And the model can successfully predict the phenotypes of more than 250 known cell cycle-related strains. And these 59 species I've talked about before, they correspond to 29 unique genes. So we took that model, and then we simulated all the mutants up to 4 gene knockout combinations. And as we were simulating, we also recorded their viabilities. So in this figure, a knockout is represented by the gene name and followed by the Greek letter delta. So in the bottom row, we have all the single gene knockouts. In the second row, we have all the double gene knockouts and so on. And in this figure, the green knockouts are predicted viable yeast mutant strains, and the red ones are inviable. We then search for this data for rescued mutants. In this figure, we have this inviable double deletion, CLIN3 BCK2, which can be rescued by additionally removing another gene from this system, which is Wi-Fi in this case. So we can look at all such examples of rescued mutants, and we focused on rescued mutants because converting an inviable strain to a viable strain happens only when we combine mutations corresponding to genes that usually have opposite functions. So we prioritized all predictions of these rescued mutants for our experimental validation. Excuse me. And then we considered each such mutant to be a target mutant. By that I mean we want to make that in an experimental setup. And our goal is to make and characterize as many target mutants as possible. And we want to then characterize the phenotypes of all these strains we produced and then use it for model validation. Crossplan is the algorithm we developed to solve this problem. But before I describe Crossplan, let me quickly tell you about the generic workflow that we use to make a mutant in an experimental setup. So genetic cross is the basis of our strategy. In a genetic cross, we can combine two given mutant to obtain a third mutant. And in this case, we can combine AC to obtain two single gene deletions A and C to obtain a double deletion AC. And in this process, due to Mendelian inheritance, we also obtain the two single gene deletions and the wild type strain. But for the sake of clarity, I won't be showing those arrows for the next couple of slides. So as we can see, cross can produce one or more mutant. And we can now take this double mutant and cross it with another single mutant and produce a triple mutant, and also make the other double mutant combinations in that process. So in EAST, the model which we studied as a test case, the various steps shown here in green are needed to perform a cross, then verify the genotype of the mutants, and for tetrad dissection and analysis. This whole process takes about 3 to 4 weeks. So because performing this cross takes a few weeks, we can perform multiple such crosses in parallel. So we define a batch of crosses, batch of S crosses performed at any given time. We define the set of S crosses that we are performing in parallel as a single batch. And as you can see, we can perform multiple such batches, one after another. And however, these batches have some constraints on them. For example, for the crosses we are performing in batch 1, you can only cross mutants that are already available in lab. For cross in batch 2, we can either use those mutants or use mutants that we've just made in batch 1. And in general, we can, for batch I, we can either use mutants that are already available in lab or the mutants that we have made in batches 1 to I-1. So in our cost model, we assume that each batch takes about the same time and cost. And we are now ready to define the cross-plan problem. We begin with model simulations. For each simulation, we recorded whether or not a strain is viable. We have identified a set of mutants that are mutant strains that are already available in the lab as the source mutants, and we have looked at some of informative predictions, in our case, rescue mutants, as our target mutants. So we want to get to these target mutants using these source mutants by performing genetic crosses in batches. So we are also given this maximum number of crosses we can make per batch. And we want to plan for k such batches where k depends on the experimental budget you really have. So our output looks like this. We have k batches of sizes S each. And once we have this output, we can actually count the number of target mutants we are making across all these batches. And our objective really is to maximize the target mutants we can make across all these batches. There are several conflicting considerations when we want to identify these batches. So for example, when it comes to crosses, there can be different strategies for making the same mutant. And we've also looked at an example where each cross can produce multiple mutants. And we should also avoid crossing mutants that are predicted to be inviable. And also, each gene deletion requires a unique genetic marker that you need to incorporate in your plan. And when it comes to batches, as you can see, there were dependencies between batches. And we would ideally like each batch to contain S crosses. And we want to maximize the number of target mutants we can make in K batches. And also, there may be delays between when we make a cross and when we can use that resulting mutant for another cross. So we developed an algorithm considering all these constraints imposed in this batch planning problem and solved it using an integer linear programming problem. And we solved this ILP using a Gurobi optimizer. So before I discuss the results we obtained using CrossPlan, I'll quickly tell you the data sets we have used in our test case. So we took that ODE-based model of budding yeast cell cycle, and we simulated up to all 4 gene knockouts in the yeast cell cycle model. And as you can see, there are 14 rescued mutants, which rescue some of these single inviable mutants. And in total, there were about 700 rescued mutants out of 11,000 viable mutants that were obtained up to 4 gene knockout combinations. And let me quickly remind you that our aim is really not to make and characterize all 50,000 odd mutants. We really want to maximize, or we really want to make as many as these target mutants as possible. So our target set, like I said, contains 700 mutants that are predicted to be rescued. And for a source set, we took 35 single gene deletions. And the batch size we have considered is 12, because we can manipulate and analyze tetrads up to 12 crosses within 3 weeks, during which the spores remain viable. And we solved this cross-plan ILP for each K from 1, 2, 3, and so on. So now I'll show one of our results. So I want to really show how the number of target mutants we can make as we increase K varies. So on the x-axis, we have the number of planned batches. And on the y-axis, we have the number of mutants we make. So when we are planning this experiments for just a single batch, all the mutants we'll be making are these double rescued mutants. So in this case, I've shown an example of a cross involving an inviable mutant. What really is happening there is either you can make this inviable mutant as a temperature-sensitive mutant, or you can genetically transform the single mutant with this other inviable mutant to obtain a double mutant. So when we plan for more number of batches, for example, when we look at 2 batches, you can see there are around 30 target mutants we can make, and know that we're only crossing, we're only making 24 different crosses at the step. So what's really surprising is that we can, in fact, make multiple target mutants from a single cross, as you can see in that image. And while we're also making these target mutants, we can also make some of these other inviable mutants that are being rescued. And we have also accounted for the number of intermediate mutants that we make in order to make any of these target mutants. But that list of intermediate mutants doesn't really count the available mutants we have. And due to the nature of these crosses, we also have certain other mutants which are not really used in any of these genetic crosses, and neither are they any of the target mutants. So when we planned for up to K equal to 12, as just an arbitrary example, we see that we could make up to more than 1.5 times the number of crosses we planned, and the total number of target mutants we made were around 200 for 12 batches. And we can also see that the number of inviable mutants we make also increases linearly here. So we have also looked at certain other extensions to CrossPlan. Like I said previously, that each of the genes in your gene knockout should contain a unique deletion marker. So we went ahead and enforced that constraint as well. And the only crosses we perform under this approach are between mutants with different markers. And we solved this problem for when each of the single gene mutant was allowed to have 4 different markers. And the maximum number of target mutants we could obtain were approximately 95% of the original problem, which did not consider markers. The number is slightly lower due to the additional constraint that we need to have different markers, which forces the algorithm to make some more intermediate mutants. And we have also looked at encoding some certain other experimental conditions. So just to summarize, our long-term goal is really to scale up and accelerate this process of building mathematical models of protein regulatory networks. So towards this goal, we have introduced this problem of computationally synthesizing experimental plans to produce a large number of target mutants using genetic cross experiments. And crossplan is the method, and it's an ILP-based algorithm, which stands for integer linear programming, to design these experiment plans. And we studied one application of this method to make as many rescued mutants as possible from our ODE-based budding yeast cell cycle model. In summary, crossplan is also highly flexible.
We're going to have to, I think, move on to acknowledgements. We're running quite a bit over. This is the last slide. Sorry about it.
So we can use this method to potentially test predictions from Boolean or logical or rule-based models. And we can define target mutants in any way we want. And the experimental workflow is very generic and can be adapted to other organisms. So I'd like to thank our collaborators at Colorado State University and also at Virginia Tech, and also the funding agencies for the grants. Thank you. Any questions?
Thank you, and we will take questions during the lunch break if you can stick around. Thanks.